Today I'm going to talk about this lunar landscape, which is really how the placenta attaches to the uterine wall. And I think it remains to this day one of the great biological mysteries, especially in humans. So I thought I'd say just a little bit about how this all works and some of the cell types that I'll be talking about. So the enlargement here is of the square to the left. And in humans, cytotrophoblasts leave the chorionic villi and they invade the uterine wall, where in human pregnancy, these cells line the mother's blood vessels. They fill up her uterine lining, interacting with the decidual cells. And in normal pregnancy, they go as far as the first third of the myometrium. So I think it's a surprise to most people, especially a lot of people who have been pregnant, that at one time your uterus was mostly filled with cells of the baby. You think about the uterus being filled with the baby, but the uterine wall is actually primarily trophoblast cells. And I think this arterial invasion is something we've studied for years. So the trophoblast cell in humans has two fates. It can become this cancer-like cell that invades the uterus, or it can fuse to form this multinucleate cell, which is in direct contact with maternal blood, and that's the exchange epithelium. That's how you get things from maternal blood into the baby. So we have studied the placenta in my group for a long time, and only pretty recently, one of the members of our group said, you know, I think I can demonstrate this anatomy by just taking a segment of a placenta that's called a cotyledon and dissect it all out and show its relationship to the blood vessel. And so that's what's been done here. This is a spiral artery, and you can see now why it's called a spiral artery, because it does spiral. And this is the segment of the placenta that it's feeding. And you can see this relationship, which is really why we're all here in this room today, is because the placenta learned how to do this. So we have been involved in one of the NIH roadmap projects. So I say this by way of introduction, that the work I'm presenting today is part of a big team of investigators. So we have been profiling the human trophoblast epigenome in pregnancy. And I think the things we've found out so far very much have surprised us, and so I hope you'll find them interesting too. So I won't say anything much about this, except that by way of introduction, we did bisulfite-seq analyses to profile the methylation. And we did ChIP-seq on 5 histone modifications, and we did RNA-seq, so we could make some statements about functionality of the modifications that we saw. And I'm certainly not going to try to show you all this data, but I'll give you some highlights. So we started out in this effort, and we looked at 5 different kinds of samples. Purified cytotrophoblasts, that progenitor cell type that I said could either form invasive cells or syncytium. chorionic villi, which are the organoids where the cytotrophoblasts and syncytiotrophoblasts reside. Basal plate, which is this area here where the trophoblast cells invade the uterine wall. And we've started also looking at the fetal membranes. And this is another area in the extraembryonic compartment where I think a lot of work needs to be done. It's usually fairly uniformly ignored. We take the placentas and throw away the membranes, but we've stopped doing that now. And so we have the amnion and the chorion, and we did not do any purification of these cells. So there's no culturing. We wanted to try to capture the epigenome in a state that was as close as possible. So even with the support of the roadmap, we were not able to do the entire gestational range we wanted to do. We ended up doing second trimester and term. So we have yet, I've just written grants to do first trimester for reasons you'll see soon, but we haven't done early gestation. And for all the data I will show you, has at least 2 to 7 samples per group. And so we're making statements based on quite a large sample number for this kind of study, but it's still small. So the first thing we did, or the first thing I'm going to show you, is our bisulfide-seq data. So the green is our second trimester placenta samples, and the yellow is the third. And right away you can see, if we do principal component analysis, that the methylation pattern of the placenta and fetal membrane changes dramatically over gestation. And you can see that the one area that does not fit that pattern of gestational age changes is the smooth chorion, which the second and third trimester samples cluster together. And the reason we think that's true is because we found an unusual population of progenitor cells in this area. Now, I can talk about this PCA for a long time, but I think I'll just point out some things. One is that human embryonic stem cells are not on the same planet as the extra embryonic membranes. You can see how far they segregate away. We can also see that colon tumor, which is one of the most hypomethylated tissues that was examined by the roadmap, also does not segregate with any of the samples that we sequenced. And we wanted to do another human fetal tissue in case what we were seeing was a fetal specific signature rather than an extra embryonic specific signature. And you can see that fetal brain also, this was a second trimester sample, does not cluster with any of our samples. So what was going on here? The tracks here are from the bisulfide-seq, and here we see, this is UCSF4, a human embryonic stem cell line that we derived in our lab. And you can see the BMP4 treated human ES cells, Jamie Thompson's model, and those cells are almost completely methylated. But if you look at the trophoblast cells, which are the next two tracks, you can see the profound hypomethylation of the human cytotrophoblast cells. And that's even when we compare their methylation with tumors such as glioblastoma and colon tumors that are known to be very significantly hypomethylated. So from this data, and here is genome-wide for our UCSF4 cells, BMP4 treated trophoblasts, the human ES cell model of trophoblasts, second trimester cytotrophoblasts, purified but not cultured cells and term cells, that you can see genome-wide that there are these profound valleys of hypomethylation. So for any of you who know about trophoblasts, people have thought for a very long time that the placental genome was hypomethylated, but I think our finding, which is new, is the extent of hypomethylation that we've discovered. And I won't belabor this, but you can look at this. This hypomethylation with the sample types down below here covers all elements of the genome. So we have not found any exclusionary areas. It includes line signs, promoters, 3', 5' elements. It's throughout the genome, and we haven't been able to find any areas that were specifically either methylated or devoid of methylation. It appears to be genome-wide. The other thing that we found was denoted by the PCA, and this is averaged level of DNA methylation. And what we're finding is that pregnancy progresses, the trophoblast genome is being methylated. And it's being methylated to a very significant degree. So it starts out hypomethylated and then becomes methylated across gestation. So we're also looking at our ChIP-seq data. And this ChIP-seq data is from the histones that I show down here. And what we found was that if you look at the histone modifications, and here we have H3K27, that among the ChIP-seq data, if a hypomethylated region, excuse me, was occupied by H3K27, then transcription was repressed. So in our cells, to a very significant degree, we think that this histone modification is solely responsible for controlling gene expression, which I think is quite new. So now if we ask where this histone modification is, then you can see a really interesting pattern. So on the top, we have trimethyl K9, and you can see in this sample that histone modification, and here we're showing chromosome 2, resides in these deeply hypomethylated valleys. So we think that this is the explanation for the repression of transcription. So in the placenta, it appears that histones play a very significant role, especially this particular histone, in controlling gene expression. And I didn't want to show too many tracks, but you can see in the expanded version of these data at the bottom, you can see this overlapping, which I think is quite interesting. And you can see these features genome-wide. So you can go in and look anywhere. We showed chromosome 2 here just because of followed 1, but we can show anywhere in the genome. So we began taking some of the ChIP-seq antibodies and looking at gestational age regulation. And we were doing this to do a developmental series, and we're not expecting to find really significant changes in these histone modifications over gestation. But what we found was really the opposite. So as you can see on the labels at the top of this diagram, CTB stands for cytotrophoblasts. And the gestational ages, GA here, show you the weeks of the tissue. And what you can see is that the histone modifications that we had done ChIP-seq for are being significantly lost over gestation. So I think the H3K9 is being lost between the second and third trimester. The trimethyl K27 is lost at the beginning of the second trimester. And when I say lost, this is obviously immunoblotting data. So this is all relative. We can still see a ChIP-seq signal, but in relative terms, there's a very significant reduction of the expression of these histones relative to the H3 loading. And here's just the quantification of these data. And the other, this is trimethyl K4. You can see this similar pattern. And this is the activating mark K27 acetylation, which I want to point out specifically to you because I'm going to talk later on about this histone modification in preeclampsia. And the quantitation is again shown to the right. And everything is relative to the H3, total H3 loading control. So we really did a lot to ask ourselves, is this really true? Because we found these very profound changes. So we did a lot of immunolocalization also using these antibodies. And I'm not going to show you all these pictures because I think people sort of glaze over after a while, but let me just show you the kind of confirmation data we have. So this is trimethyl K9. These are floating villi. These are the chorionic villi that are bathed in maternal blood. And you can see the histone staining here is very much reduced during pregnancy. These are the gestational ages, 6, 19, and 39. These anchoring villi are the ones that give off these invasive-like cells that enter the uterus. And the anchoring villi, you can also see the same sort of picture that the cytotrophoblasts that have been launched into the uterus also show this pattern. So we don't think what we're seeing is an artifact due to our isolation of the cells. we can see it in tissue sections of the maternal-fetal interface. And we've asked lots of different kinds of questions with these data, and I'm not going to belabor all of this. You know, we've obviously done a huge amount of pathway analysis, but I thought I would show you one thing that I find interesting. Now, the code down here, the first letter, if you look down at the bottom here, the U means unmethylated inhuman ES cells. The next letter in the 3-letter code is for second trimester cytotrophoblasts, and the third letter is for term trophoblasts. And you can just see with our methylation data alone, so this is methylation data, if we looked at what was unmethylated in human ES cells and then methylated in trophoblast cells, then we could bioinformatically pull out the pluripotency factors. If you look at all these, we got some of the greatest hits here. If we look at what's methylated in human ES cells and unmethylated in trophoblast cells, then we get things that we know are very interesting trophoblast factors. Transcription factors that we know play a role in development of the placenta. And as Miriam said, we have worked on oxygen, the role of oxygen in placental development for a long time. And we see that these elements are highly unmethylated in trophoblast cells. And then we're really interested in what happens at term. And we think that the AP1 elements and the HIF elements are, again, playing an important role. So we've gotten lots of interesting testable hypotheses out of these kinds of data. So our lab has worked on preeclampsia for a long time. And I'll tell you a little bit about preeclampsia, and then before I show you our data, I'll give you some caveats of what I'm going to tell you about. So preeclampsia is a pregnancy condition. The statistics worldwide are that about 8% of women who are pregnant for the first time will have some form of this disease. And it's called preeclampsia because preeclampsia in Greek means sudden or lightning. And it's been recognized for a long time because women since recorded time, when they were pregnant, they would suddenly die. And the cause was usually preeclampsia, so it was so dramatic that it was named by the Greeks. And what happens in preeclampsia? Well, you get this sudden increase in blood pressure. So women who have had normal blood pressure their entire pregnancies, all of a sudden have this very sudden, very dangerous increase in blood pressure. And they have many signs of maternal vascular damage. They have edema, which is one sign. They have headache. Their vascular system is in a lot of ways showing a lot of elements of endothelial damage. Excuse me. And another sign of this is spilling protein into the urine. So what happens in preeclampsia? Well, I showed you that first photo where the spiral artery was feeding the placenta, and here I show you the opposite of that. So we have a cotyledon of the placenta, and the placenta, the blood flow is not getting to these tongues of placenta that it should. And if you do a cross-section of the maternal-fetal interface, this huge spiral artery that should be blown up is not expanded by this process of trophoblast invasion. Now I want to comment here, the SPE here, it means severe preeclampsia. So severe preeclampsia, it doesn't sound like a specific medical diagnosis, but it is. So it's a very specific kind of preeclampsia that happens before 34 weeks of pregnancy. The blood pressures are higher, the symptoms are worse than in regular preeclampsia that might happen at term. It's the most serious form of the disease because it can happen so early that it threatens the outcome of the pregnancy. So not only the mother, but the baby's life is threatened. So we are studying only severe preeclampsia in our cases. So one of the things that we noticed was the fact if we took our RNA-seq data, which I'm not showing you in any detail here, but if we took our RNA-seq data and looked at the genes that were regulated between second trimester and term, so we're looking at what could be the transcriptional consequences of this very large shift in the epigenome. When we looked at this, you can see the pathway analyses here. You can see a lot of genes are regulated. But what we found that was very interesting, we thought, was many of the genes that we found that normally should be downregulated between second trimester and term were in our gene sets that were upregulated in cytotrophoblasts and preeclampsia. So I'll say this again, and I'll turn it around backwards. So if you've looked at any gene sets of purified trophoblast cells in preeclampsia, you will notice a really interesting phenomena, which we were never, ever able to explain. And that is most of the genes in the preeclampsia trophoblasts were upregulated, which is so odd because when you look at array work, you're used to saying, well, some are up, some are down. And in our preeclampsia genes, they were always up. And from our RNA-seq work here, looking at what changes between the second and third trimester, we found that many of our preeclampsia genes, which are on the top here, were ones that were supposed to be downregulated normally at term. So I hope that's clear to people from what I've said. Okay, so then we decided to use our same approach and ask, if we use an immunoblotting approach, can we see big changes in histones that accompany the development of preeclampsia? So here we see now immunoblotting, and we've used preterm labor. So NPTL in my blots, and our blots means preterm labor, but at the N is for non-infected. Usually one of the biggest causes of preterm labor is infection. And so these are women who had preterm labor due to some sort of structural abnormality or some unknown cause. And when we looked at gestation match samples and did immunoblotting for the histone modifications, we found in preeclampsia that we got some differences for sure, but we got a huge upregulation of acetylated K27, which is an activating mark, which we reasoned could be one of the reasons that our gene expression data always showed that in cytotrophoblasts, the gene expression was always up rather than the mixture of up and down you would predict. So here again is immunolocalization, and you can see that this mark, here's a case again of severe preeclampsia, the early dangerous kind, and you can see by immunolocalization in the trophoblast cells, which are shown by their expression of this mark, you can see that it's highly upregulated. So this is just taken out of the placenta and nothing done to the cells. So finally, we asked the question of where this modification was ending up. So here are our samples from second trimester, and here are samples from term. And this is ChIP-seq for K27 acetylation. So the first thing I would point out about this data is you can see this loss of modified sites between second trimester and term that you would have predicted by our immunoblot data. And you can see the numbers of sites. There are huge numbers of sites involved in this. And we certainly don't think this is the only sort of histone modification change that's happening over gestation. The other thing now I'll point out is our preeclampsia data. So these are all individual samples of purified cytotrophoblasts from women with severe preeclampsia. And you can see in most of the cases, we have a module, a very large module of modified sites that are in common with second trimester and not being downregulated as they should be as advancing gestation. And I would point out that is in almost all of our preeclampsia data, we have an outlier here. So I think people who work on this pregnancy complication will find this very familiar, that one of the samples doesn't fit the pattern. And so this is pretty much what we always suspect. And I'll show you what these data look like. Here are preeclampsia samples. And this is from a region of chromosome 19 that contains genes that are specific to the human placenta. They're called placenta-specific proteins. They're down here. There are a whole bunch of them, and people still aren't really sure what they do. They might play a role in metabolism, in growth. We just don't know. And there are also some other really interesting genes in here, some cell adhesion genes and some chemokines. But if you look at the second trimester tracks from this region versus the third trimester, you'll see that throughout this whole region and through a lot of the genome, the preeclampsia samples are retaining the second trimester pattern. And so these pregnancy-specific genes are among those that we find are consistently upregulated in preeclampsia, and many of our colleagues have found the same thing. So the conclusions that we've drawn from this study are that the DNA of primary cytotrophoblasts, and in fact the whole extraembryonic compartment, I dropped all that other data as I talked to you, but many of the findings, in fact, if not all of them, apply to the entire extraembryonic compartment. The hypomethylated regions are occupied, the deep valleys are occupied by trimethyl K9, which I showed you repressed gene expression. And we think if we do enough samples, this will also be true for trimethyl K27. And we showed for K9 that occupancy of the hypomethylated regions with this trimethyl K9 repressed transcription of over 1,000 genes. The cytotrophoblasts, we're very interested in the concept that this unusual epigenome plays a role in establishing cytotrophoblast fate. I think one of the things that's always been really interesting to me is how can you get such a functional trophoblast cell in humans 5 or 6 days after fertilization by launching transcription factor pathways. You need to have this very sophisticated cell that knows how to attach to the uterus and start launching its adhesion programs and its immune programs. We're really interested that this is a component of this could be determined at the epigenome level. There is a tremendous gestational age regulation of the placental epigenome. And we're viewing this in the context of the fact that the placenta has a 9-month lifestyle, a 9-month lifespan. And we're really interested in whether or not this could be a feature of aging in other cells. So my colleagues at UCSF who study aging are now looking at some of these questions. We're also interested in the fact that this unusual epigenome and regulation of gene transcription, does this confer particular vulnerabilities? And I think so far we've mostly looked at preeclampsia, where we found this big change in K27 acetylation occupancy that confers an early gestation fingerprint to the genome, which we think is one of the problems now in preeclampsia. But I would hasten to say we have looked at several other pregnancy complications, and we're also seeing not the same exact ones, but perturbation of the epigenome. But we think that this unusual epigenome and the perturbations that could it might undergo could be a basic cause of many of the pregnancy complications that we as humans experience. So I wanted to credit the huge number of people who have been involved in this project. Olga Gemenchev, who's worked with me for years and years, and our whole team of people here at UCSF helped prepare the samples and analyze the data. My colleague at UCSF, Joe Costello, who's a cancer biologist, listened to me bang his ear for years and years and years about how cool the placenta was. And we wrote an application for the roadmap, and it got funded. A lot of the basic elements and fundamental parts of the data analysis were done by Ting Wang and his colleagues at WashU in St. Louis. And Bo took the lead on this, so I particularly want to credit Bo Zhang, who has done yeoman's work on this for sure. And the data were generated by Martin Hurst and his colleagues at the British Columbia Cancer Center. And I'd be happy to answer any questions.